we actually had lots of fun. I have chemistry students from analytical chemistry, chemical biology, material chemistry, and even recently I have organic chemists to join us. I have biology students, you know, from pharmacology, pathology, and you know, molecular biology programs. So they will utilize these newly developed technologies to address biological questions. And I have MD students and some MD PhD students in the lab, helping us connecting from the basics research to the clinical research. But everyone in the lab, regardless they're chemistry students or biology students or MD PhD students, they all have to learn mass spectrometry. Okay, so over the years, because of my strong, you know, industrial roots, we have grown a very strong partnership between academic and the industry. And all based on the roots of ultra high resolution mass spectrometry, we now grow many branches of the tree. So we're fascinated about developing these omics technology for precision medicine because we believe that this is really powerful to decipher the complexity of biological system and understand disease mechanism at the organ level and the cellular and the molecular level. So our goal is to integrate the omics, mainly proteomics, metabolomics, with the in vitro and in vivo functions to better understand the disease mechanism and also for better diagnosis. So as you know, we trained by Fundamental Clafferty, so top-down is in my genes. So we pretty much started to do top-down when I started, you know, as a graduate student. So, but why top-down? Obviously, we know like unlike the genome, which is a blueprint, you know, static, but the proteome is very complex. There's one gene can be corresponding to many protein products due to those alternative splicing isoforms and post-translational modifications. We have Ben here, so Ben is expert on the post-translational modifications. So essentially, the proteome brings us closer to the real-life building blocks. But it is very difficult to study post-translational modifications by traditional biological techniques such as Western blot. So mass spectrometry is certainly one of the most powerful techniques to study post-translational modifications. And also in the top-down approach, we analyze whole proteins. So we can show, you know, the post-translational modifications together with genetic mutations and alternative splicing isoform. So what this can be used for to solve biological questions. Typically, as you know, that for the inherited disease, there's certain mutations, but how this mutation causes disease is not known. So in those top-down, we can see how those mutations will cause post-translational modification changes, etc., and possibly in fact affect their protein-protein interactions. So these are the major instruments that we acquired recently, and that's the workhorse for our proteomics and metabolomics efforts. And we want to thank NIH, the S10 grant for the purchase of these instruments. So as we said, the ultra high resolution mass spectrometry is so powerful. So when I was showed the biologists, they said, you know, you look at your gel, it's a protein band, you thought it's pure. And when we show them, and there's so many different peaks, I said, don't get a heart attack because it doesn't mean that your protein is not pure. It just means that there are all sorts of post-translational modifications, like the acetylations, phosphorylations, and also degradations. So the proteins can be degraded, but they can still be phosphorylated. And for some proteins, you can see those oxidations as well. So in this case, there are over 30 different proteoforms from a single gene products. And then the question is, how are we going to use these PTM for? Okay, so we try to see whether we can detect those PTM, you know, reproducibly. Again, Ben's lab has great examples on the histone modifications. Here I show you the example on this cardiac troponin I, which is a gold standard biomarker. So what we find is this troponin I, they are in different cardiac disease developmental stage. There's a reduction, consistent reduction of this phosphorylation with the severity of the disease. So by looking at these different level of these post-translational modifications will allow us to gain a better picture of different disease progression stage. And thanks to the high resolution mass spectrometry, that we can show this difference between acetylation and trimethylation. So many times the biologists will ask us, oh, can you, identify difference of 14 Dalton, like methylation? We said, okay, of course. So you actually can detect this differences with 14 Daltons, easily, and even with 0.036 Dalton mass differences between acetylation and trimethylation. Again, I mentioned so much about the Bay here, but Garcia's lab also showed Orbitrap can be used to differentiate these minor differences. Our lab using FTICR, so we find this protein called regulatory light chain. It expressed in atria, it has acetylation, versus when it is expressed in ventricle, it has a trimethylation. So obviously, they have different functions in the atria and ventricle. So we did publish this paper in Cardiovascular Journal. So we also said it seems top-down is easy approach, right? From you just look at the whole proteins, but in reality, everybody knows top-down is quite challenging because top means measuring the intermolecular mass. So it's like you climbing a mountain. Getting to the top isn't easy. The down means fragmenting the interproteins. Just try to do fragmentation, use all kinds of fragmentation techniques, CAD, ECD, ETD, VPD, etc. But fragmentation, we said getting down isn't easy too. So for those you probably try to ski, what you need to get to the top of the mountain is get a lift, right? And if you have good skills, skiing skills, you can getting down the slope quite easily. So what do we say? The lift means we need to develop tools. We need to develop all kinds of top-down proteomics tools to help make it much easier for others to perform top-down experiments. And the skills means train students. So the trick is, I always said, getting students when they're first year, don't do bottom-up, get them to do top-down so they don't have any fear factors. And they would think, oh, okay, top-down is pretty easy. And that's because from my own experience, when I started my graduate school in Phys Lab, we are not allowed to do bottom-up. So we naturally get used to do top-down proteomics experiments. And for top-down proteomics, it has 3 parts. It's a front-end sample preparation, separation of interproteins, the middle part, top-down MS, analysis of interproteins, and the back-end informatics. Okay, so in the past about 8 years, so we have been working on this, developing these novel approaches to address the challenges of top-down proteomics. There are several challenges. We are focused on these 4. So first is solubility. Proteins, generally speaking, very difficult to be solubilized, especially those membrane proteins. And 1/3 of proteome are actually membrane proteins. So the biology labs always using SDS. It's like the gold currency in the biological community. But mass spectrometrists don't like SDS. Why? Because once you have any little trace of SDS or triton or any of these conventional detergent or solvent, we got a beautiful spectrum of the surfactant, not your mass spectrometry, okay? So this means we cannot use these conventional surfactant. So we are developing new top-down MS-compatible surfactant in the lab. And we do have some success recently. As you know, the proteome complex have millions or even billions of proteoforms. So we really need to separate the proteins first before we get to any mass spectrometer. Even FTICR do not have enough separation power to separate, millions, billions of proteoforms directly by mass spectrometry. So we have been developing the novel multidimensional chromatography for interprotein separation. But proteome is so dynamic. There's 10 to 12 orders of magnitude differences. So even you have used multiple separation technologies, you still cannot detect those low abundant proteins. Okay, so this we're trying to develop these nanomaterials to mimic antibodies to reach those low abundant proteins and PTMs. We also try to facilitate in these data interpretation. When I was first studied in medicine, so that's not really a good and user-friendly and also free software. We try to develop this mesh software. Right now, we do have those versions available freely to the community. Okay. So we have worked on this photo-cleavable surfactant for almost 6 years before I finally get it work. So we made a synthesis to make those cleavable surfactant by inserting photo-cleavable linker between this hydrophobic tail and hydrophilic head. Okay, so then we use them to solubilize the protein. Before mass spectrometry, we use the UV to degrade them. They're very fast. You can see this, the degradation can occur in about a minute. So then this will not interfere with mass spectrometry. As you can see, these Azo, we call it Azo. This stands for 4-hexylphenol azosulfonate that has a very high solubilization power, similarly as SDS. But it shows that in the mass spectrometry, when you have even 0.1% of SDS, you do not see and detect any proteins. But when we have the Azo in there, we can still see the signal. And DDM is a popular native mass spec surfactant. But you can see DDM is not very powerful in solubilizing proteins. So overall, we have shown that Azo not only have very strong solubilization power, but it also is mass spectrometry compatible. So then we use the Azo to solubilize, we call it the toughest degrees, which is the part of the very difficult to be solubilized. We deplete the soluble proteins first, and then you show these insoluble palette. So you add Azo versus the time you don't add it. So if you have Azo, apparently it can solubilize those nearly insoluble proteins. And my student Kyle Brown led this project. He found after depletion of the soluble proteins and assisted by Azo, he can recover many of the mitochondrial proteins, and this is from heart tissue. So he identified those proteins in the electron transport chain. And very interestingly, we finally get the top down of this phospholambian, which is a very critical cardiac proteins. And phospholambian is too small for bottom up, because it's only 6 kilodalton, but it's too hydrophobic protein for top down. But with the assistive Azo, we finally were achieved the high resolution spectrum for phospholambian and locate the sites for palmitoylation. We also recovered the whole protein complexes. This is ATP synthesis. So responding to a reviewer asked us, he said, can you use the Azo to run SDS page, mimic? So we actually find out you can use Azo completely to replace SDS, and the gel looks pretty good high resolution. So this shows that broad impact beyond proteomics. And recently we find that the Azo not only works for top down, it actually works even better for bottom up. So it helps in two folds. So it helps solubilizing protein in the bottom up approach, and it also helps with digestion. So we can digest everything pretty much in this half an hour. Okay, so we use the FASP as a benchmark, and we find there's no difference between 30 minutes and overnight digestion. So, and we then extended this Azo-enabled strategy to extracellular matrix, which is like very difficult to chew bone, because these proteins like collagen, proteoglycans, etc., are nearly insoluble. And with the assistance of Azo, we're able to solubilize the proteins, even like multiple component of collagens. Okay, now I'm going to switch gear to talk a little bit about the chromatography strategy that we have been developing in the lab. So unlike peptide, proteins have a much more diverse range of physical chemical properties. So this gives us opportunity to develop multidimensional chromatography incorporating mutually orthogonal mode. Okay, so we have this called, let me actually show the pointer. So most people in the LCMS know that reverse phase chromatography. When we talk about LCMS, they thought about reverse phase, but there are several other chromatography mode, even just based on polarity. For example, the hydrophilic interaction chromatography. Again, this is pioneered by Ben's lab. They separate histones. My lab works a lot on this hydrophobic interaction chromatography. We also try to couple with ion exchange chromatography and SEC. Okay, so why are we so much interested in hydrophobic interaction chromatography? Because HIC is hydrophobic interaction chromatography, provides a high resolution separation of interproteins under non-denaturing conditions, so it can retain the protein structure and function. But traditionally, because the use of a high concentration of non-volatile salt, high in the heptameter series, such as ammonium sulfate and sodium sulfate, rendering HIC incompatible with direct MS analysis. So my student, Bifan Chen, in collaboration with Andy Alpert from PolyLC, they developed the new series of more hydrophobic HIC material that can retain the proteins, use MS-compatible concentration ammonium acetate, which are the buffers that native mass spectrometers use. So the HIC, very nice feature of HIC is it's very sensitive to post-translational modifications. So remember, as post-translational modification could potentially alter the protein structure and conformation. They could be separated pretty nicely in the chromatography mode in using HIC. So then we find that this has a really great usage for monoclonal antibody separation. This, I don't know how many people here from pharmaceutical industry. I see some of you works in Janssen, Johnson & Johnson. So maybe for the antibody drug conjugates, really is rising as a very important biologic for therapeutics. So we have been collaborating with AbbVie, so we kind of separated nicely with these antibody, monoclonal antibodies by HIC. And we can see on the monomers, dimers, trimers, that's resembling the native mass spectrometry. We also can show the different glycosylation forms. And this actually data was acquired in Maxis QTOF. We're pretty excited about the high accuracy. Also can achieve those, deglycosylation and separate them under mass spectrometry in the HIC MS. So you can see both the glycosylated form and also deglycosylated form. Okay, so using FTICR with ECD, we are able to achieve a good fragmentation of the intact monoclonal antibody and probe this drug binding region. Okay, so for many people, like to work with peptides because a tritide has an advantage of high signal-to-noise ratio in the mass spectrometry. As Kelleher's lab, has published a nice paper in analytical chemistry, there's a combined effect isotope and charge state on signal-to-noise and function protein mass. So essentially, there is an exponential decay of the signal-to-noise with increasing of the molecular weight. Okay, so this means you have to separate low molecular weight proteins before you can detect the high molecular weight species. And this is also explain why most people work with peptide. Okay, so as Kelleher and Neil's lab actually pioneered this gel-free system that can separate proteins. My lab has taken a chromatography approach. We developed this high resolution size exclusion chromatography by combine set protein and SECs with different pore size together, and we achieved pretty good resolution with these 3-column combination. So again, if we only use one-dimensional chromatography, you can see most proteins are relatively lower molecular weight species, less than 50 kilodalton. We did get a very nice peak separation. But when we use an SEC at the front before the reverse phase, this allows us to detect many high molecular weight species. Many proteins about 60 kilodalton and 116 kilodalton, even up to 223 kilodalton and 140 kilodalton proteins. These are very, very important cardiac proteins that we are interested. Okay, so now the question about these are more qualitative, determination and analysis. In order to bridge with biology analysis, we need to demonstrate the reproducibility in the protein, quantitation. So my lab has established this method that we show that you can quantify the protein expression level and also modification simultaneously. And we did this linear curve, a standard curve generation, and we find if we can separate this small subproteoform, subproteome, about 20 to 30 proteins, we're able to separate them well, and they do have a linear response. So, and then we show that the direct comparison of those proteins, from the top-down LC-MS, label-free, with the Western blot, they're quite consistent. Okay, so now getting to another challenge in the top-down proteomics is the dynamic range. So we do need to enrich those low abundance proteins. So we have been developing these nanoparticles. The nanoparticles has comparable nanometer size to antibodies. So it has high capturing capacity due to high surface area. It also has multivalency, so it allows high binding affinities, similar to antibodies. And we also use that to highly specific capture of proteins and PTM by those ligands, these affinity binders. So our first success has been used to this, generation of these functionalized cobalt ferrite nanoparticle with online LC-MS for top-down phosphoproteomics. So this generation of these iron oxide and also this newly cobalt ferrite nanoparticles, they can enrich, bind to the phosphoprotein strongly, and then we can elute and do LC-MS. And this is a loading mixture. So as you can see in the loading mixture, there's not many of phosphoproteins. But after we enrich with nanomaterials, this elution that significantly enrich the phosphoproteins, allow us to perform online LC-MS to determine the phosphorylation site. And recently, we've been very interested in, of course, to detect the troponin I. This is my favorite molecules from blood. But as you know, in the blood, there's, as I said, extremely challenging because of a highly abundant albumin, right? So we all know albumin is the dominating protein in the blood. It has over almost 10 orders of magnitude than those tissue leakage proteins such as cardiotroponic I. So cardiotroponic I is a biomarker for heart disease because it's supposed to stay in the heart in the healthy condition. So when the heart is injured, it gets released into the blood. So you can detect them in the blood. That's why it is called tissue leakage proteins. So they leaked from tissue during the disease, and that's why they serve as a biomarker. So we try to use this nanoproteomics approach, again, use this, engineer this troponin I affinity binders and modify the nanoparticle surface. So we can then enrich troponin I, different, various proteoform of troponin I. And as I said, detected by QTOF mass spectrometer. And we not only detect this naked, unphosphorylated, but also phosphorylated, doubly phosphorylated, and also degraded form. You can see the high accuracy we see, we actually obtained for this based on these actually impact to data. So comparison to antibody, actually our nanoparticle will be able to enrich more diverse proteoform platform than the antibody enrichment. So recently we published the work and shows that the nanoproteomic strategy is very effective in enriching those low valence cardiotroponic I's from the blood. So, and to our surprise, that our nanoparticle performs better than antibody because it completely depletes, nearly completely depletes the albumins from the blood while it enriches the troponin I. And how that would be useful, because so these are the troponin I's, we spike the blood and they enrich them by nanoparticles. The top two panel from healthy patient, this is the middle two is from diseased patients, and on the bottom actually from postmortem sample. So you can clearly see those molecular signature being detected by high resolution mass spectrometry after enrichment of the, from the blood. Okay, so we really excited about this results because, as you know, detecting proteins from blood is really almost like a nightmare. So we'll be able to see the molecular signature that could potentially differentiate the patients into different subgroups for better treatment. And so this project was led by material chemistry students and actually chem bio students, but both of them now are mass spectrometry experts in the lab. And this shows you the traditional, this is ELISA assay. So previously you determine whether patient has heart attack or not, all based on these color changes. And now we show this to, some, even the patients say, well, this is too crude assay, right? To your whole life, or death is determined on this very crude assay. So we definitely say we really want to develop this comprehensive troponin assay to better diagnosis. This is because this current ELISA assay, it has pretty good, exclude the patient who actually don't have a heart attack. But there are so many patients with all elevated cardiotroponic I, but only a portion of them actually, after this hospitalization, cardiogram, CT scan catheterization, they be released from hospital. This is the data we got from our UW hospital, okay. There's more than 40% of patients actually do not have an attack, but they have to undergo all these very invasive techniques. And so this is a lot of money wasted there, because each patient to go through those individual procedures costs about $100,000 to $200,000. So we definitely hope to allow the better diagnosis of these majority patients that has elevated troponin but may or may not have heart disease, okay. So this is where we want our nanoproteomics assay to come in to help differentiate those patients better. And this essentially, we use this for precision medicine because there are so many different causes for troponin elevation. Based on the ELISA, just color change is not enough. So we hope using the PTM as next generation biomarkers. And we are now trying to implement a large scale clinical study. Okay, so now talk about the software that we have developed. Remember, many people do not feel comfortable or confident to do top-down proteomics, not because they don't have the instrument. I mean, Orbitrap now is pretty popular and the QTOFs. Many people has the difficulty of getting softwares, because first of all, you probably don't want to spend a lot of money to buy a software when you just started. So we have now developed this Mesh Explorer, a comprehensive user-friendly software for top-down proteomics. It can process data from various vendor formats. So it also incorporates multiple algorithms for deconvolution and data research. And it is totally free. So we, thanks to the kind of reviewers that we got this software grant funded, and we really wanted to reward, put the effort to serve the community. So if you want to use this software, please contact our team. We still have 2 years more funding. We will be able to provide the best support to you as much as you need. So again, we recently published this paper and this also shows there are so many people around the world as using the software. We also have our we try to do our user group meetings on a quarterly basis. Okay, I don't know how much time I have. So I'm going to briefly talk to you about my biological programs, which is just going to be a quick overview. I know I don't want to keep you guys too long in staying here. My research mainly focused on studying heart failure, because this is disease for both men and women. And when I started, I'm trying to get funding also from cancer, but they all turned me down. But cardiovascular community welcomed me, with open arms. So that's why I started to study cardiac disease, and I'm just getting more and more interested in this area. So we use both the large animal models and the human clinical samples to study heart failure. And first, I focused on the sarcomere as monofilament protein. When I first submitted my R1 to the cardiac panel, and they said, when I talked to them, I want to sequence the entire proteome of the sarcomere. They said, well, that's too ambitious. So then they asked me to focus on a few proteins to study. And that's why I get started. And then we started to establish this top-down quantitative proteomics platform. We extract proteins from a minimal amount of cardiac tissue samples. And they can do tissue homogenization, protein extraction, and run LC-MS. And then by this online LC-MS rapid profiling, we can immediately detect the potential alterations in the disease stage in comparison healthy stage. And our lab also can, remember, a lot of mass spectrometers are pretty sensitive. So we can collect large fractions for offline MS characterization. You can also use these for functional study. And finally, we can correlate the post-translational modifications with disease. So when we first developed this model, this is a acute myocardial infarction model in pigs to mimic a human heart attack. And so we basically exclude the left anterior descending aorta for 90 minutes to induce infarction. And so when we making sure there's definitely infarction occurs, and we take those tissue samples and do mass spectrometry. So we find that as changes in those troponin I in this acute myocardial infarction model, again, there's a decrease of phosphorylation. But interestingly, there's another protein that also shows a significant reduction of phosphorylation, and we later identified this protein as enigma homolog isoform 2. So again, when you have a lot of opportunity and ability to detect post-translational modifications, a lot of time we try to ask, how can we use these information for, right? Can we show any for potential therapeutic development? So we actually showed this here, these altered post-translational modifications by myocardial infarction. Okay, so this is a reduction of the phosphorylation of these very important proteins we discovered, ENH2 and troponin I. So the previous model we actually used in the Wisconsin pigs. So this is in collaboration with my longtime collaborator in Alabama. Actually, you see this completely reproduced our data. There is decreased phosphorylation in the troponin I and ENH2. But amazingly, after they treated with stem cell therapy, with the heart functions improved, we actually see these molecular level of reversal of these we call more adaptive post-translational modification changes. So it reverses these post-translational modifications, and also support the fact that these stem cell therapy could affect the cell signaling. Okay, so I'm just quickly going to show you a recent study that we have with this hypertrophy cardiomyopathy. Remember, we said the top-down that we will use as they can show post-translational modifications together with protein mutations and alternate splicing isoforms. So I thought it's really going to be useful to study this genetic disease. For example, this HCM, hypertrophic cardiomyopathy, it's caused by genetic mutations at proteins in the sarcomere. But it's so diverse, right? There's so many different causes that requires precision medicine. And to make things worse, the genotype is not clinically useful in predicting the prognosis in the HCM, because some of the mutations associated with relatively benign clinical course are not manifesting late in the life, but other mutations leads to a more malignant clinical course and died in early stage. And even patients with the same mutation can exhibit different disease severity. So I literally talked to a cardiologist. He said, oh, I'm so difficult, in position to give patient potential treatment options, because even he diagnosed with this mutation, but they could have be okay or not okay. So that both choices are open. So that means we really need to look at the proteome level. So we have the opportunity to work with cardiac surgeons, receive those samples from septomarctomy surgery. Because remember, this hypertrophy cardiomyopathy patient have very thickened heart muscle, and they eventually block this blood circulation. So to the stage, if this surgery has to be done, or else they cannot survive. Okay, so we got those small piece of this heart tissue from the septomarctomy surgery, and to our surprise, we find that even there are different genetic mutation, but when patients all exhibit similar phenotypes at the septomarctomy surgery stage, the proteoform changes are very, very consistent. So our data actually suggests the common pathways are actually activated to precipitate the similar phenotype in HCM patient. This shows that the therapy for treating those HCM patients should not be dependent on the patient genotype, but rather dependent on what we call proteoform type or the phenotypes. Okay, also we have something interesting to share is we find there's a consistent phosphorylation sites in the human and the pig model. So that shows pig heart is actually quite translational model to present human models. Obviously, you cannot using human to study early stage disease. So the pig is giving us really great opportunity. So also to make sure you can study as the patient, individual patient level. Again, heart disease patient don't want to give you a piece of their heart when they're not too sick, need a new heart, or need a septomarctomy surgery. So how are we going to study that kind of patient? We use this iPS cell derived cardiomyocytes. So from individual patient, we derive those iPS cells and differentiate them into cardiomyocytes, and this is a multi-lab collaboration. And we can do these 3D engineering cardiac tissue. Okay, so we can actually grow those cells into 2D culture or 3D engineered cardiac tissue to study both proteins at the protein expression level and also PTM level. And we have established unbiased proteomics method to assess the maturation of these human pluripotent stem cell derived cardiomyocytes. And here are the cells actually grown by my students. That this is a ECT, 3D engineered tissue culture, tissue. And we can use them for precision medicine. You can study for regeneration, cardiac regeneration. You can use them for disease modeling. Those of you are pharmaceutical companies, right? You really want to test the drug in the individual patients before actually getting to the clinical trials. So while we hope to decode the human heart proteome by top-down proteomics, we have developed a good method for the sarcomere, right? So now we are interested in membrane proteins because we now have the Azo. So we are, some of my students are studying the receptors, and we also interested in the kinases and phosphatases. Okay, so overall, we really tried to integrate the proteomics, metabolomics with in vitro, in vivo functions and clinical cardiology for cardiac system biology. This is still a long journey ahead, but we have lots of fun. And of course, we show that the top-down, it's additional D here, proteomics that can be used for precision medicine. We take the patients, collect these the tissues, the blood, the bodily fluids. We can do proteomics on these clinical samples that essentially we use in protein PTM and expression changes as integrated biomarkers that to segregate patient into different groups that we can use for more precise and personalized treatment. Okay, I really have to thank my group. I have many very talented graduate students and of course the postdocs to help with this research. They did a lot of work, and thanks to funding and also collaborators. And I'd like to thank you so much for your attention. I know it's pretty close to your dinner time. Hopefully you enjoy the talk, and I'd be happy to answer any questions you have.
Thanks a lot, Ying. Great talk.
Sure, thank you.
I'll open it up for questions. If anybody would like to go ahead and start, you can feel free to go ahead and unmute yourself and ask away.
Should I stop sharing so we can just talk, or? Any questions?
I have a question. This is Harsha calling from J&J. So you mentioned your MASH software. What is the deconvolution algorithm that is in MASH?
Oh, that's a great question. We have so many different algorithms. So we worked with, Thrash deconvolution algorithm. And I think recently we also tried to employ, as I said, the MS-DConv from Xiaowen Liu and, lab. So, I think even the recent UNIDAC by Michael Marty. So they all really want to collaborate with us. And the very recent publication in, I think in Cell Reports, or anyway, so they also have this fast deconv. So our what we developed essentially is an interface, right? So we are not developing these unique deconvolution software or searching algorithms. Okay, here you go. So we have multiple deconvolution algorithm, MS-DConv, Top-FD, eThrash, PPAS-TD, and ProMax. And as I said, recently UNIDAC will be included as well. So it is a platform. So essentially we can incorporate all the newly developed algorithm into the same interface so that the users.
I have a question regarding the differences in the algorithms, because in pharmaceuticals, we are evaluating various algorithms to deconvolute our antibodies and our therapeutics, right? And we get varying answers based on the type of deconvolution you do. So in your experience, using different, algorithms to deconvolute the data, you might definitely get differences in certain types of proteoforms, or even certain types of lower level, especially the low level glycoform.
Right.
So what is your experience in using these different algorithms to kind of, is there some way of unifying the answers together, or you go with one over the other? How do you determine that?
So we actually recently has published a paper in JASMAS to show in honoring the John Ye's, distinguished scientist award. So we have shown this as a called ensemble to a machine learning strategy that can couple all these different algorithms together and have a uniform output to address the question you have.
Yeah, so, in terms of error rates, right? I mean, is there some way we can, eliminate some of these things that are biased towards certain features in your data?
Yes.
Than,
right. So, yeah, especially if you're looking at like drug binding, right? So glycosylation. So this program, of course, has. Yeah, so the very nice feature is it's a visual, so you can get into to really annotate them or validate manually, right? And you can use all these different algorithms, deconvolution, and you can compare those results. Let me show you whether I have the. Sorry, I don't have it in here right now, but yeah.
I got your point. I can the references. Yeah.
Yeah, I'll try to find those references for you. See later if others also will be interested to see that. Actually, I mentioned about this. This is actually by Sean McAveen. He's a computer scientist working on this project. Yeah.
Yeah, I have one final question. In terms of, you were looking at troponin as your main protein and looking at post-translational modifications, phosphorylation mostly. Is there some way that you can use this to stratify the patient populations into subgroups based on disease severity or any other type of, maybe other type of comorbidity, or like disease
Right.
state or, it's very complex when you talk about biomarkers, right? Your
Right.
diet can influence the level of, glycosylation that's on a protein.
Right.
Yeah.
No, absolutely. So you asked the question. So the Troponin assay, as I mentioned, right now it is a good this called a rule in rule out method for the ELISA. Just by looking at the expression level changes is not enough, because a lot of patient has elevated level. They may or may not have heart disease. So there are very diverse range of causes for the troponin elevation. That is not enough to be diagnosed based on the ELISA assay, which is very, very crude, right? So this is why we say that potentially those PTM fingerprints could allow us to segregate the patients into different populations. You're right, maybe comorbidities, likely with diabetes, with hypertension. There's also, I think there's also kidney disease known to cause some kind of troponin elevation. And the stringent exercise could also cause, troponin elevation. So, but we cannot say that it could be used as a marker yet until we have demonstrated in the large human cohort. So we are trying to establish a big clinical study. Unfortunately, because of COVID, so it's very difficult to recruit patients, and we cannot really collect any blood samples from patient at this time, yeah. But definitely we're hoping to working with pharmaceutical companies, because interestingly, a lot of pharma, we interested in troponin assay because all the drugs, and before they can be approved by FDA, they have to test this cardiotoxicity, right? So, and troponin assay is used to assess whether this drug has a toxic, in the cardiac effect. And many very potential potency drugs cannot pass because the troponin elevation in the ELISA assay. But actually we show that the ELISA assay, those antibodies, there are lots of problem in those antibodies because they have batch to batch variation, hydrophilic antibody interference, et cetera, right? So they're just, and there's lack of standardization. So I'm talking to several, pharma right now, they say they need a better, comprehensive, accurate assay so that don't trash their drugs so quickly, right? So this is another real important usage. As I said, quantitation of those post-translational modifications from tissues and blood now is possible. Of course, as I said, Ben Garcia's lab has done so much for the quantitation of the histone modification for epigenetics. Yeah, I assume a lot of people are from Ben's lab here, right?
Thank you.
Ben has done wonderful work in the proteomics quantitation of PTMs, histones, epigenetics.
Great. Does anybody else have any other questions? Feel free to unmute yourself or send them in the chat box.
This is Jing from Vela. Can you hear me?
Hi, how you doing, Jing?
Good. How are you?
Good.
Yeah, actually, I read several papers published from your group. Actually.
Oh, thank you.
Especially the method development for the intact top-down using the separation and collaboration with Andrew.
Yeah.
Andy, and also later on I found the paper about the Azo.
Yeah,
that's very helpful. Actually, I work in the manufacturing biologics. For us, we need to separate different kind of variants to ensure the quality of the drug. I do see that the top-down method could streamline our quality control in a much better way. But meantime, we realized that the top-down, we don't have so many tools, as you said.
Yeah.
Right. So we don't have chromatography methods. So I thought your group did some effort to try to build up some using some new material. That's very helpful. So I want to know, like, is that possible, like from like expert, like in your field, can provide some guidance in the kind of like to help people build a top-down method using Higg or multimode, chromatography to separate different variants based on like a protein size, charge. You just, if you have a protein, you know some properties, you follow this kind of guide, so we can get an easier way to build a method to save time to convert to practical method.
Oh, sure. I think we are working with AbbVie as just this model, right? So we are actually helping them developing the method and publish a paper. You see, I think we published 3 or 4 papers together with AbbVie. So they have this collaboration program that support one of my students to work on the project. So actually we have very strong collaboration with pharma. So they basically, because also my background, in pharma.
Yeah, I think I saw that.
So, yeah, so we actually definitely help the farmers develop a method and then help them translate the method into their samples. I mean, so they will give us some more of a, standard protein, standard antibody drug conjugate to develop a method. And then we then translate the method to them to more of a proprietary compounds. Is that what you asked?
Yeah, I understand.
Okay.
You're working pretty good.
Yeah, we will be happy to collaborate with you.
Yeah. Actually, the multi-mode column, actually, I was also, learned from your paper quite a lot, yeah.
Yeah, no, we also, establish, as I said, lots of potential collaborations to strengthen the, as I said, interaction between academic and industry.
Yeah, okay. I have a question for the biomarkers you talk about later on part. I'm not in that field, but I'm just curious. So in the conventional, like a bottom-up method, is that possible to do the same thing, or this can be only from top-down method for
the biomarker for so I think of course you can still use MRM, SM to detect the specific modification, right? Okay. But you would not just be able to observe we call the molecular fingerprints easily.
Okay, different level of phosphorylation, is that, or?
Yeah, so you cannot see the whole picture of the proteins. You see, you might be able to detect this certain site of modification, right? But you cannot take a look at the whole molecule, like whether it has phosphorylated, it has acetylated, etc.
Yeah, I do agree. Actually, top down is actually close to the real,
right?
The real situation.
Right, but if you want to say just monitor the specific site changes, modification changes, again, as I said, you have the local expert. Garcia's group has developed many of those great middle down, bottom up, those site-specific modification quantitation method, right? But the thing from us is that this allows you to get the whole picture, especially in the drug discovery stage, right? So you want to see different modifications, right? So that's what we think is most useful. Like in here, whoops. That this kind of fingerprinting, you will not be able to achieve using bottom-up approach.
Okay. Question, like for this one compared to ELISA, how comparable the sensitivity? I mean, not especially for this protein. I mean, probably in the future, you envision, is that possible? Maybe, top-down can replace ELISA.
Yeah, so that's a great question. The problem right now is the sensitivity of mass spec are not comparable with ELISA assay yet. So the ELISA assay, I think they can get into 0.04 nanogram per mil. We are the LOD about 0.5 nanogram per mil. So it's actually better than we thought. We originally thought it was even, we could not even achieve that level. And the 0.5 nanogram per mil, it's already in the clinically useful range, because a lot of patients actually has elevated cardiotroponic I. They are in this range already. So this assay right now can be applied to the clinical samples, but it cannot detect those very, very low amount of troponin I elevation yet. So this instrument we use Maxis to. So we are hoping this timsTOF can get a better sensitivity, but right now the LC that is built for the timsTOF is not optimized for top-down. It's optimized for bottom-up. So a lot of time you have good instrument, but because you don't have good LC system, so that cannot be used for top-down. A lot of mass spectrometers are really made easy for top-down now, but chromatography is the main problem actually.
Yeah, I see the point. Still,
yeah,
some way to go. A lot of way
to
go in the future. Yeah, to solve problems. Yeah.
Right. So come back to the question previously by Asha that we do have this paper published to address to your question. So we have enhanced these analysis by combining different deconvolution results through the machine learning strategy that can help solve the problem that you have. And ultimately, we are hoping this is provide a uniform, standardized software platform that, so to solve a lot of questions about, different vendor instrument, you can come out with different results and different algorithm, in the deconvolution or search, they give you different answer, right? So that's very challenging. So that's actually part of the reason why we got this grant is we try to provide a standardized platform to process all vendor format data.
Yeah,
so
definitely. Yeah, I'm going to take a look at that paper, but also like in the list of algorithms that you had, MaxEnt1 or 3 or the parsimonious algorithm that's been written by Proteometrics are very, very popular in pharma, especially for antibody characterization.
Which one? You say, sorry.
MaxEnt1, maximum entropy.
Oh, the maximum entropy, yes. So
there's also the parsimonious deconvolution algorithm that Proteometrics has developed, which actually resembles the mass, like the charge state envelope of on the raw data. There's no artifacts in the deconvolution and things like that. So so that's very, very useful for us to kind of evaluate these things, especially because we are trying to use these different vendors and they don't really provide a complete solution because they try to promote their own.
Right. That you have a great question. So we actually cannot incorporate those, because those like MaxQuant or other software that they sell for price, they will not give us the algorithm for free. Right? Remember that our software is free. And so we do not have ProSite, the algorithm, but we have the Thresh, of course. So, but I think the ProMax, they're getting better. The UniDAC really is made more for a good deconvolution. So there's a lot of free, open access, open source, software.
Can these algorithms handle ion mobility data? Can you get ion mobility resolved deconvolution of your data?
We are continue to add those features on, right? So right now, Bruker, we got Bruker timsTOF, so we have to modify the software to upgrade it to handle the, the ion mobility data. So again, if you can try the mesh, and as a user, you can give us suggestions to improve, right? So again, we still have 2 years of funding, so it's a good time for you to raise up any, you know, needs you want, right?
Yeah.
Yeah, we have a wonderful team. You know,
Yeah.
they're really excellent. Sean, as I said, they're all computer scientists who are helping mass spectrometrists. Yeah.
Thank you.
Yeah. So thank you. Good question.
Ying, this is Sanjeev here.
Hi. How you doing?
Good. Hey, sorry, are we finishing up, or do I have a chance to ask you one question?
Oh, okay. Sure, Sanjay. How you doing?
So my question is very good work. Actually, very impressive.
Oh, thank you.
My question to you is, like, you know, you're talking about sensitivity is very low for intact proteins. So what instrument are you using for those purposes?
Oh, yeah. We now use Bruker instrument right now.
Is it the FT instrument or?
No, QTOF works really well for
QTOF. Wow.
For top-down. Yeah. So if you so all the data, most of the data we collected actually from QTOF. So I have to tell you that honestly, our lower end instrument is mostly used before the timsTOF joins. So the Impact 2 gave great results, you know, top-down data. And also the MXS gave beautiful data too. timsTOF works great for direct infusion, antibody drug conjugate. We can even resolve the whole antibody drug conjugate in timsTOF. But as I said, right now, the embedded LC coupled default LC doesn't work for LC-MS mode of top-down proteomics. We're still working on it. You know, FTICR is useful if you want to have ultra high resolution for proteins. But again, you know, those data, a lot of data I can show you are actually from TOF. Like this study. These are from Maxis, so it's you know, you can get these easily. This is actually all Maxis data, Max QTOF. So QTOF works beautifully for interprotein analysis.
So what concentration are we talking about here?
So I think this is online approach. I have to check, as I said, I don't work in the lab right now, so I have to check.
No.
But it's actually very little amount needed, almost under a few hundred nanogram range of proteins.
Very nice, very nice.
Yeah. So yeah, this is from FT. But FT, you know, if you want to get really good resolution, FT is great, right? You can also use ECD. But my point is a lot of data we acquired, like this is also from, I think this data is from Maxis too. Maxis, it has a pretty good resolution up to 40, you know, 42 kilodalton. You can see resolving power. Yeah, it was pretty good.
Yeah, that looks
see
very
here.
good.
And this is 22 kilodalton proteins. I come back to Harsha. The deconvolution software, Max is really good deconvolution software, but they probably won't allow us to incorporate in the mesh because you know, then they cannot sell the software, right?
Yeah, I mean, I wouldn't say Maxent is the best because sometimes you get a lot of deconvolution artifacts as well as artificial peak sharpening, and that could be really problematic sometimes in a pharmaceutical setting where you have to kind of potentially probably rank orders,
yeah, you have the right
based on your modification levels, and
I know
if you get the right answers, you'll be really off, right? So
so sometimes a deconvolution seems like too magic, too good to be true, like this deconvolution, right? So I mean, it looks beautiful, but you know, how much kind of a data, you know, manipulation in the process, we don't know, right? So, but you're right, you know, you need multiple algorithm to double check.
Nice.
Yeah, but those, yeah, Sanjeev, so these data all come from Maxis.
Okay, thank you.
These are not from FT.
Okay.
And yeah, so though all these data actually these are from impact 2. So we found impact 2 is more reproducible even. So
I see.
my group of biologists, biology students from my group all love the impact 2 because it's much more robust.
I see.
Yeah, and it's very, can provide linear quantitation.
Very interesting. Thank you.
Yeah, sure.
I know we're running a few moments late, but does anybody else have any other questions?
Okay, see.
All right, well,
Okay.
Ying,
Sure, I'm trying to stop sharing.
all righty.
Okay, so great talking to you guys.
Yeah, thanks so much for taking the time. We really appreciate it.
Yep.
Okay.
Bye-bye.
Thanks everyone for joining.
Enjoy your dinner.
All right. See you, everyone. Take care.
Sure.